particles that can flow by ways of simple diffusion. Now, if we look at our paperwork, over here on the second page of the little first handout, where it talks about diffusion being defined, and then you guys remember how I said that I didn't have a space there, and it needed to have a space, and it says simple diffusion involves mostly non-polar substances like oxygen, carbon dioxide, lipids, hydrocarbons. They move straight through the phospholipid bilayer without the need for proteins. That's telling me something such as oxygen, which would be getting transported in my blood on a red blood cell. Is everybody following me? Okay, so if I've got my little red blood cell in here, and on that red blood cell, it's carrying oxygen. That's O2. I know I'm not drawing big enough to be seen back there at the back. But let's say that the oxygen level is higher in my blood than right here at my tissue level. That would get to diffuse. Does everybody see that? So oxygen would get to diffuse. My cells that need it would get it. And it moved from an area of high concentration to low. It moved down its concentration gradient. Does that make sense? Okay. The picture that they're showing here is just simply if you drop like a salt cube or a sugar cube into water, you get to see it just simply diffuse through the water. But I kind of like our stuff about the body because that's what we're dealing with. We're dealing with the body. The term viscosity. Have you ever heard somebody talk about how viscous a substance might be? In what terms? Probably thinking about biology. A lot of people tend to think about cars because they talk about oil for the car, and this is dealing with, you know how oils have different weights, like 5W, 10W, the 10W30, that sort of thing. That's letting you know the weights of that oil, and then it kind of tells people who know stuff about cars how viscous that fluid is going to be, meaning allowing the engine parts to move freely. So, let's kind of take that idea and apply it to the human body. What would we need in our human body to flow easily? Blood. Blood. If you're not well hydrated, it's harder for the blood to flow. If you're well hydrated, blood flows easier. Not as much strain on the heart, which is why it's important to drink water. Not soda, not tea, not coffee, none of that, okay? Water. Because the better the amount of water in your blood, the more easily it flows and the less strain it puts on your heart. So diffusion, movement of molecules across the plasma membrane. The ones we just mentioned, oxygen, carbon dioxide. They simply moved across the membrane itself, the phospholipids. That one was termed simple. When we need a protein to move that substance, it's termed facilitated. So I think that was the next term on the paper. And that one means those particles are polar. For example, glucose. Glucose is the simplest sugar. And it's the one the cell can use first. However, cells do need to control its movement. So the way to do that is to have a protein that will facilitate the movement of only the glucose. So now, even though we're going mostly from high to low concentration, it is still a way for the cell to control what moves into it. No energy, still no energy. We don't need energy to do this. And it's helping to control the amount of glucose that might move in. We mentioned oxygen, CO2, that sort of thing. Those get to move straight through those phospholipids. But that's good. Deprive a cell of oxygen, you're going to kill it. Deprive a cell of getting rid of the waste from using oxygen, which is CO2, you're going to kill it. So letting them move just through those phospholipids is good. So we have our simple diffusion and we have our facilitated, which would be the protein. For the movement of water, osmosis. This is where water gets to move across that plasma membrane, goes from areas of low concentration of solute to high. On your paperwork, just underneath diffusion, we had osmosis. Once again, note, passive process, no energy required. Water wants to go. Note that it's telling us, okay? Water wants to go from an area of the lower concentration of solute across the plasma membrane to the area of higher concentration of solute. This does create the osmotic pressure, the force on the cells. Under normal conditions for animal cells, we want materials simply doing this number, back and forth, back and forth. Inside the cell to outside the cell, inside, outside, inside, outside, inside, outside. This helps us create a condition of pressure that is isoosmotic. Iso meaning same. Therefore, outside the cell would be the same as inside the cell. We are not putting any pressure on the cell. Now let me explain why that is important to animal cells. We are animal cells. We do not have a cell wall that could protect osmotic pressure problems. Animal cells simply have the plasma membrane, which means we're squishy. We have to make sure fluids do not put a pressure, meaning that fluids would push that cell in on itself, or fluids that can make that cell expand. We need our cells to exist in isotonic conditions. So, if we get to have isotonic conditions, materials are simply doing this number back and forth, back and forth. The cell is happy, the tissue is happy, blood is happy, okay? And there's no pressure on any of those cells. Any situation other than this puts the pressure on the cell, and the cell can die. If we put an animal cell into a solution, meaning the outside of it is more than what's on the inside of the cell, because the way things like to move, we would be seeing materials move in a fashion, and it would kill the cell. So, do I have a picture with the cells? There we go. Okay, so if we take an animal cell, if we put it into a solution that has too much outside of the inside of that cell, materials will get to move out. That animal cell crenates. Now, I think on one of your other the second handout that I gave you, I don't know what I did with my copy. You have that term. Or it might be on the first one. Y'all help me out here because I got y'all reading. Yes. Okay, it's on the first one. See where it starts talking about tonicity? Okay. So, this very first one, if we have isotonic, both the cell and the solution are the same. Movement is good. We've got stuff flowing back and forth. No pressure on the cell. If we look at a cell that is put into hypertonic, where the solution is greater than inside the cell, notice that it says osmotic pressure gradients pull water out of the cell, and the cell shrinks or crenates. All right? The cell will shrink or crenate, C-R-E-N-A-T-E. That cell's dead. So this means that if a person's salt levels, for example, are incorrect in their body, they could be killing off cells of tissues. And that needs to be corrected quickly. Now, if the opposite happens, and we place a cell into a hypotonic solution, meaning we don't have as much in the solution as we do inside the cell, Water